Hello, everybody. I'm Dr. Carmen Puliafito, and I'm pleased to be here today with Dr. Karl Csaky. Welcome to Retina Synthesis, Karl.
Carmen, looking forward, and thank you so much for inviting me.
Karl is CEO of the Retina Institute, Retina Foundation of the Southwest, where he is also director of the T. Boone Pickens Laboratory for Retinal and Macular Degeneration Research. Karl is an accomplished clinician scientist who very recently co-founded a company, Vinci Pharmaceuticals, which is looking at some innovative delivery systems and drugs for retinal targets. And one of the first areas of interest is dry AMD. So Karl, what is your vision for treating dry AMD as we move forward?
Yeah, so it's one of the big last unmet needs, right? And it's a tough nut to crack because things move slowly and the degenerations progress slowly. So unlike DME or macular neovascularization, where we can see results pretty quickly, it's a little more challenging in the area of dry AMD. But the hope is that as we start to understand the impact the disease has on various anatomic features and functional features that we didn't really appreciate even 10 years ago, that hopefully we'll be able to identify precursor lesions, lesions that put patients at higher risk for progression, and hopefully be able to then use therapies to slow progressions and or potentially improve, in some cases, some functional aspects of the disease. So it's going to be interesting next several years as more and more folks are getting into the space. Of course, we've been in the geographic atrophy space for a while with, unfortunately, some notable failures, but we'll see what happens in the next couple of years.
Intermediate AMD is a real challenge because patients have visual impairments. They come to us complaining about them, but we obviously don't have a therapeutic advance other than the ARADS vitamin protocol. So what are your thoughts about measuring and endpoints in intermediate AMD? And then most importantly, when would you consider intervening in intermediate?
So I think, that's one of the things that I was fortunate when I was at the NIH. I was able to work with Wiley and his team and, really try to get into a dialogue and a conversation. Because as we all know, when we're thinking about a therapy for any disease, there are numerous hurdles that you have to think about, right? You have to think about, first and foremost, are you really going to have an impact on the disease pathogenesis? And then, of course, if you have kind of deciphered what you think is going to be the pathway and you want to, somehow interfere with that, then you've got to be able to design clinical trials that you can measure some degree of efficacy. And then, of course, that has to go to the FDA, and the FDA then has to review it and come up with some, terms of approvability. And then, of course, you've got to then go to insurance carriers and worry about reimbursements. And then lastly, you have to worry about, folks in the trenches, the docs, and whether or not they'll be willing to use the therapies. So I think with something like intermediate AMD, you've really got to think of all of those things in aggregate. So you can try to identify, it's clear that there are a significant number of our patients who don't get worse. They stay stable for many, many, many years. They have, for the most part, minimal deficits. And as I tell them, you don't really need anything. We're going to watch you, but you're not showing any signs of change. But clearly there's a group of patients, and as I said, we're starting to identify OCT signatures of those patients who we think are going to progress more rapidly to atrophic changes. There's no argument that the development of atrophy and the expansion of atrophy are approvable endpoints, right? So the Wiley and his team have made it very clear to us that the loss of photoreceptors is a bad thing. So if we can identify patients who even have intermediate AMD who are at high risk for losing photoreceptors, either through assessing their functional deficits or through more OCT findings that we're seeing now precede these changes, I think we have a chance. So I think there's been a lot of development over even the last 5 years in our understanding of how to decipher and split apart this large group of patients that we call intermediate AMD, although Wiley in his last discussion kind of doesn't like to use that term, but clearly there is a group of patients that are at higher risk for progression.
What is your startup's technology and approach to this problem?
Right. So, this is an area that I've been working on, for a long, long time. And, I was trained, my PhD is in pharmacology. I've always been interested in understanding ocular pharmacology. And I've always told people that I think of all the things that we do in the clinic in terms of retinal diseases and trying to understand what can get into the clinic, understanding the ocular pharmacology of a therapeutic is one of the most important things. And so one of the things that I've been working on for many, many, many years is this idea that what are better ways to get drugs into the eye from outside the eye, right? So all of our therapies, the majority of our therapies right now are administered through needles in the eye or through implants in the eye. And while that overcomes lots of the barriers, right? These barriers that we face, it becomes more challenging when you think of patients who still have relatively good vision, who might progress at slower rates. And so you really want to be able to investigate a therapy that is minimally invasive, that can deliver an agent over many, many months, potentially. And so our therapy, so the device that we are developing is an episcleral drug delivery device. We've optimized certain features that are overcoming, we think, some of the barriers that prevent drugs from getting into the eye. Some of the work that we've done over the many, many years has shown that the clearance of drugs away from the eye is very robust. And so by kind of isolating our API from those clearance mechanisms, the hope is that we can retain drug levels on the surface for longer periods of time. And at the same time, we can then alter the API in a way that we think also optimizes its ability to get into the tissue. And of course, the placement of this device more posterior gets it closer to the tissue, the retina, and especially the retinal pigment epithelium, and even the choroid, which we now are starting to identify as a major kind of source of disease in AMD. So the hope is that this approach will allow us to deliver, our first API, but then going forward will be relatively API agnostic. So you could potentially put any API or therapeutic in this device once we've optimized and we know kind of how to control the delivery to the tissues. So that's that's our approach.
What's the molecule of interest to begin with?
Right. So right now, we are big fans of the concept of mitochondrial dysfunction in AMD. Mm-hmm. And we still think that plays a pivotal role. Complement even interacts with mitochondrial in many significant ways. So the source, we think, of a significant pathology is that the mitochondrial dysfunction. And we know that some of the agents that can cause, the improvement in mitochondrial function, and we've shown that in the lab, is glutathione, naturally occurring agent that are at reduced levels in patients with AMD. And so the API is essentially an esterified form of a precursor to glutathione. The concept of esterification, it's been around for a long time, and latanoprost and there are other agents that have definitely are on the market, and they simply use the concept of esterification to get drug, more efficiently into the eye. So what we've done is esterified a group of precursor APIs that will be delivered from this device, and that will allow us to deliver potentially delivers the therapeutic for many months, because we can of course load the device and and get a sustained delivery kind of aspect to the patients.
What would a phase 1 trial of this device and drug look like?
Yeah, I mean, I think, like is kind of the norm. You always want to be thinking about, the safety first. One of the things that, we were very much focusing on, and as you know, a lot of what kind of kills approaches is any kind of safety index. And of course, the precursors to glutathione are extremely safe. We've already had some experience, and of course, we place devices in the episcleral space all the time, buckles and all kinds of things. So we know that that space is pretty tolerant to anything. So while we're, proposing, a safety study per se, we're going to try to see if we can get some degree of early efficacy signal, even from the phase 1 study.
So what kind of characteristics would you be looking for in patients in that?
Yeah, I mean, I think, what we begin to see is that this group of patients who have already are either at high risk for developing GA, or who have some extrafoveal GA, or even those patients, interestingly, who have significant changes either with drusen or reticular pseudodrusen, but most importantly have significant low luminance deficits. And we've been able to measure low luminance deficits not only in visual acuity, but in using a novel automated quantitative contrast tool that also measures low luminance contrast. So we've identified, you can identify groups of patients who are at significant, who exhibit significant deficits with these tools. And so the hope is that as you assess the safety of this device in these patients, we might see, if we're lucky, some signs of improvement.
So low luminance reading, do you think that's a potential endpoint that the FDA might look at?
Yeah, I mean, I think, in when we held our 2016 symposium that I helped run, the FDA, and this is true for many years, they're very much supportive of functional endpoints. I mean, that's ultimately what patients want, is they want functional improvements, right? And so any functional assessment is something that they look on very favorably. The challenge becomes that, as you know, even with visual acuity, right? Visual acuity is a wonderful endpoint. It's used to approving, lots of our agents, but there's a high bar, right? I mean, there's 15-letter gains that you have to demonstrate. Why? Because all of our visual function tools tend to be very noisy. And so you've got to be able to demonstrate a therapeutic benefit that overrides the noise, the test-retest variability. So one of the things that we are very much, what we look at, especially with some of the newer algorithms that you can impose on these functional assessments, is you can, in some cases, dramatically reduce the noise. And so the hope is that you'll be able to generate some signal with some of these low luminance tools that we're using right now. Low luminance, reading speed is one of them, but again, these tend to be somewhat noisy, and there's lots of retest variability. So we have to be very much aware of that aspect when we go with functional assessments. But I think, there's a group of patients that I think we can identify, and we've been doing that already in the clinic, and we've started to do that in clinical trials for GA, for example.
I think that's very exciting. All of us as clinicians know that patients complain even when their Snellen acuity is not bad. They're really impaired in terms of functions of everyday life reading. So looking and developing other endpoints is really quite important, both in terms of treatment and documenting the efficacy of your interventions.
Yep. Yeah. So I think, and I agree. I mean, I think that's always been, and now that we have these tools, you can really see in some cases profound deficits. And what's most interesting, and we've published some of this work, is that they don't always correlate to the anatomic changes. So if it's just drusen alone, a drusen doesn't necessarily always correlate with these functional deficits. So there are other OCT signatures that we're looking at as well. So there's clearly a heterogeneous kind of cadre of patients, and we have to identify those patients who are most at risk and most severely affected by their AMD.
Can you talk a little bit about the episcleral implant, what it's like and how it's
it's
used?
yes. Yeah, so it's something that we've iterated for many, many years, right? And one of the things, and of course, it's always a challenge. I mean, we've seen, failures. Anacortef, is probably the most famous failure of a drug that was placed in the episcleral space, right? And there's reasons that, I was fortunate enough to work with Alcon at the time. And I think we've learned, and I think sometimes we learn more from the failures than we do from any kind of success. And I think we learned a lot about, what are the barriers, and we published a lot of that work. As I said, it's really quite profound, the speed at which drugs placed on the surface, even a sub-Tenon's injection, I mean, you've seen this, and we've all seen this in the clinic, where we inject a large bolus of, mostly triamcinolone or whatever agent we have in the sub-Tenon space, and in a very rapid period of time, the fluid is gone. So the clearance rates of fluid away from the eye are extremely rapid. And so what we've tried to do is design a device that kind of isolates the API from these very robust episcleral clearance vessels. Some of them are lymphatic, some of them are blood vessels. And so as the device then allows you for the drug to basically be isolated from these clearance mechanisms. And then hopefully, and again, the size of these, as the episcleral space can tolerate really large devices, Barvelts and Maltinos are, enormous devices. We won't need that large a device. But I think we've got enough capacity in the space to then load the drug into these devices that will give us 6 months, even a year of delivery if needed. The early phase of course, will be shorter duration, just for they're easier to think about the kind of how to organize a trial, a 3-month trial versus a 6-month or a 12-month trial. But going forward, if things look good, then we can go ahead and start delivering the drug for longer periods of time.
So what other kind of molecules can this delivery system work with?
I mean, it's really, in many cases, it's agnostic. I think the only thing that we don't have the capability of delivering at the present time, but we're working on it, and that are, proteins and RNAs and things like that. Although again, going forward, there'll be capacity potentially for that. But right now we're looking at mostly small molecules, and in our cadre of small molecules, another drug that we're looking at. And again, we're looking at pathways that we think will be more straightforward and easy. So things like even Napafinac, which is of course a non-steroidal, is something that it's not esterified, it's an amidization. So it's got an amide group, but it also has the same concept of lipophilicity, and it gets into the eye, we know from topical administration, to a small degree. And so the question is, can you get more robust tissue uptake by sustained episcleral approach? And so that's something that we're looking at. And of course, as I said, it's relatively API agnostic. So we can really start to look at a whole smorgasbord of potential mechanisms and API as we go forward, especially as we start to fully dissect the pharmacology of that space in patients.
Is the delivery system something that would be implantable in the office setting?
So, the ultimate idea, and it's, this is where, it's a very interesting discussion between, what's obviously the device could be placed potentially in the office. There's also reimbursement issues that favor placement in an ACS, for example, right? It could be a relatively simple procedure, And in some cases might be needed at the beginning as we start to make sure that the device is placed in the proper location. And of course, as you know, being full aware of all of the reimbursement dynamics that can occur, placement of this device in an ACS, for example, would be something that I think a lot of retina specialists would look at favorably in terms of both control and safety, but also for potential reimbursement as well.
Well, this seems like a very powerful technology for delivering sustained delivery of drugs to the eye in general, actually.
Well, we'll see. Like I said, I mean, there are some real challenges, and I think, all of us who enter into this, into these kinds of stuff, there are lots of unknowns and lots of challenges, but we're hoping that, as we push forward, we've got some really smart people on the team, and we've made some really good progress, both in the technologies and in animal modeling and things like that. So I think we've got a pretty good shot at hopefully getting this to work.
Well, Carl, you've always had a focus on translation in your research, but now you've taken it to the next level because you're really trying to bring it to the marketplace. Tell us something about the founding of your company, what it's been like, what have been the surprises?
Yeah, I mean, I think, when I look at medical research, right, and even now I sit on NIH study section, so I'm reviewing grants, and there's a whole, kind of spectrum of important research that's being done, everything from basic science. I think once you start to really think about translating anything into the clinic, there's so many other aspects that you have to consider. You have to consider the business model of the company, right? Is it going to make sense in terms of something that, I wasn't really aware of, cost of goods, right? So we know that there are certain things like gene therapies, for example, where the cost of goods is very high. And so that means that the costs going forward are going to be high as well. So having to worry about how many patients potentially could receive a therapy, where you want to get your kind of returns, all these business aspects, the people who are looking at the company, while they get excited about the science, and I think the science is hopefully the critical driver, there's all these other aspects that, I wasn't really fully aware of. And as I talk to people, especially on the business side of these kinds of efforts, you can really see that, their perspectives, have a degree of kind of aspects that I was not aware of. If you're in the lab all the time and you're dealing with patients and you're thinking that I've got some great ideas, there are all these other aspects that you really have to come to, understand and may influence how you eventually bring that product into a company and try to push it forward.
Do you have any advice to colleagues who are interested in pursuing a path such as this?
Yeah, I mean, I think it's, again, it all depends on, where you want to go in terms of a disease, right? So I think if you're picking something like, dry AMD, which is tough, right? I mean, there's really no, even when we look at the AREDS formulations, we know from the data that there's really very little impact on the dry component of AMD, right? It doesn't really affect drusen volume. It doesn't affect GA growth. So we really, in those kinds of aspects, you really have to kind of be prepared that there's going to be lots of twists and turns. And I think it's critical. And one thing that I'm a big fan of at any area of research is the ability to iterate. You've got to be able to iterate over and over again so that as you push forward and something doesn't work as well as you think it should work, you've got to be able to easily iterate. I think that's one crucial aspect. And the other crucial aspect is I think you have to be brutally honest with yourself, right? I mean, when we do the, when I look at some of the data that Vinci is generating, right? You have to be very, kind of brutally honest with yourself. Is this really, have we crossed the bar? Has the company crossed the bar that we think is going to be, there's going to be success at the other end of the effort. And I think that's a little bit different than the lab, right? So the lab, we're working hard, we get successes, but when you're doing a company thing, you know, you've really got to raise the bar to a higher level. So as you push forward, you know, you don't end up doing missteps, and that's going to cost you time and money.
Well, Carl, thank you so much for this conversation. I think the potential of what you're working on is extraordinary. And I think your insights into dry macular degeneration and drug delivery are really terrific. And so thanks a lot for being on Retina Synthesis. We appreciate it.
Absolutely. Carmen, enjoyed it. Be well, and thank you so much.
Yeah, great.